What we are going to explain here, the meaning of this lecture is that if you have the nanoparticle, how you could imagine that where you have to use them. So always think for some kind of useful applications where you could use them. Try to see in what way you could take the maximum advantage of the nanoparticle. So here I'll take you some of the environmental applications that how we can use the nanoparticles. It is not only the gold nanoparticle we'll be talking about, but you will have different kinds of nanoparticle. Here will be, you will see. So some other very important other kinds of silicon oxide nanoparticle, titanium oxide nanoparticle. So we'll be talking about their applications as well. Uh, This slide I showed you earlier. So here I mentioned that when we have to talk about the growth of the nanoparticle in the form of aggregated assembly, this is known as aggregated assembly. Schematically, we can show it like this. Uh, you are familiar with the nanoparticle when they are uh, present in the suspension individually placed. But when a group of nanoparticle is placed individually on a platform, that kind of assembly is known as aggregated assembly. So here what we are doing, in fact, we are growing the nanoparticles on micelle. You see, this is a very huge micelle. So you read the scale bar is 100 nanometer. Suppose if you asked me what is the size of SDS, sodium dodecyl sulfate micelle, and usually the sodium dodecyl sulfate micelle is 1 to 2 nanometer maximum. So, but in comparison to sodium dodecyl sulfate, this is the micelle which is a very big micelle, right? And the micelle, the formation of the micelle has taken place exactly the same way as in the case of sodium dodecyl sulfate. So now I'm going to tell you that this micelle is made up of polymeric molecules, not the monomeric molecules. When you have a polymeric molecule, it is a very big molecule. So therefore, polymeric molecule is expected to produce a very big micelle. So just for comparison, this is the SDS molecule. So when we have so many SDS molecule, they will aggregate in a micelle like this. So this is the micelle of SDS, okay. But on the other hand, if I am taking a polymer like, I think I have placed the picture, which polymer I am talking about, this one. If I take this polymer, you see now, this one is polyethylene oxide, CH2, ethylene, oxygen oxide, and you see the square bracket. It means that this part of the polymer is repeating how many times? 99 times. This one is polypropylene, polypropylene oxide, so it is 1, 2, and 3. Three carbon functional groups, and this unit is replacing 69 times. So you could see that this entire molecule is in the form of a very long chain where these fragments are repeating 99 times, where the polypropylene oxide is repeating 69 times. Such kind of molecule or the polymer is known as block polymer. It means that you have one block of polyethylene oxide, another block of polypropylene oxide, another block of again polyethylene oxide, and schematically we can show it like this. It is polyethylene oxide, and then again you have a block of polypropylene oxide, PPO, and again it is oxide. Now the question is, this molecule forms the micelle, but what kind of micelle and what is the shape of the micelle and how the micelles are going to be arranged. So what we do actually, the polypropylene oxide is more non-polar. It is predominantly non-polar, whereas polyethylene oxide is predominantly polar. Not entirely polar, but predominantly. I'm using the word predominantly. So I can depict this entire polymer in the form of a structure like this. So this is, you may say, PEO, PEO, and PPO, polypropylene oxide. When such molecules are polymeric molecules are going to aggregate, they are they aggregate in such a way that the predominantly non-polar part should be maximum away from the water molecule or the aqueous phase, where the predominantly polar part should be maximum closer to that of the aqueous phase. So how can I create a micelle for this? So it is like this. Sorry. So in this way, I can draw the micelle of uh, polyethylene, polypropylene, and the polypropylene units are directed in the center of the sphere of the micelle, whereas the polyethylene, they are away from, and this is now is the entire micelle. Now you can compare, this is the micelle of polymer, okay, and this is approximately, whereas this one is approximately 100 nanometer. And you could see this diagram, right? The black dots are in fact the gold nanoparticles. Now, what happens because of the ether oxygen? So here you have ether oxygen. In structure is lined with the ether oxygen like this, and there are lone pairs on the ether oxygens. Now these lone pairs can provide electron density to any electron deficient species, for example, Au plus 3. Au plus 3 is the gold salt and the gold ions, right? And what they can do, they can provide the electron density by providing 3 electrons. It can convert Au into atomic state. And once the atoms are created, the nucleation instantaneously starts, and then a nanoparticle is created. This is what shown over here. You see that, just a minute, let me see. Yeah. So this is the micelle which I have shown over here, right? So in this micelle, again, the way we explain that the conventional micelle is having the core and the stern layer, the peripheral. Also can differentiate the parts of the micelle, that detail in which we talk about for the conventional micelle, where I said that you have the core, inner palisade layer, outer palisade layer, and then the stern layer. But in this case, so it's the core, which is represented by the polypropylene oxide functional groups. Those are embedded in the interior of the sphere, and whereas the polyethylene oxide functional groups, they lining the periphery. Now you see a cavity. The cavities are created, as I mentioned over here. These are the cavities. These cavities. So the reaction is taking place. Reaction is taking place. The nucleating centers. The nucleating centers from different gold nanoparticles like this. This is a tiny nucleating center. Nucleating center, if somebody asks you, the nucleating center is the first few entities. Those when grow into full-fledged nanoparticle, and they usually possess few atoms, maybe 50 atoms, 100 atoms, or few hundred atoms. That is a nucleating center. The nucleating center itself is not a stable nanoparticle, but it is an entirety which is in the course of the growth, and hence it will eventually converts into a bigger or full-fledged nanoparticle. So this nucleating center is created here in the cavity, and it grows into a full-fledged nanoparticle. It eventually grows into a full-fledged nanoparticle. That is why these black dots are the nanoparticle, and the black dots, all black dots are the gold nanoparticle, and they have grown from mycel. So exactly the way you might have heard about the template effect. So we have a template, and we are using a template in order to design a particular object. So exactly the same way what we are going to do, we are having the micelle. Micelle acts as a template, and it helps to grow the nanoparticle. And the now nanoparticles are restricted to the surface of the micelle, and they will remain embedded in the micelle. This kind of assembly is known as a self-assembled state of the nanoparticles, right? So this is the transmission electron micrograph. If you want to see the scanning electron micrograph, this is the scanning electron micrograph. You see clearly the gold nanoparticle grown on the surface of a spherical micelle. Now, the mechanism for the growth is I have shown over here. This is the surface cavity. The reaction which I have mentioned over here, electrons provided by the ether oxygens which are over here, ether oxygens. So these ether oxygens, they have electron density because of the lone pairs, right? And these ether oxygens are providing to the gold atoms. Once gold atoms are created, the nucleating center is created. That nucleating converts eventually into a full-fledged nanoparticle. So that nanoparticle grows all over the surface cavities. So this is known as a template effect. If somebody asks you, what is the template effect? Is the way that we could put the nanoparticle which is dispersed in the aqueous phase, aqueous suspension inductively. Can we direct them on a specified platform? You will say yes, it is possible. You could allow the reaction to take place on a platform or on a template, so that all the nanoparticles which are going to be produced, they will remain on the platform that is aggregated state or self-assembled state of the nanoparticle. So we will talk about what is the advantage of this. So now you see now nanoparticles over here, nanoparticles over here. So the advantage of this, uh, I'm just going to mention the catalytic applicability of the self-assembled nanoparticle in comparison to the nanoparticles those are dispersed in the solution. So here we have two reaction vessels. The one is where the nanoparticles are dispersed in the aqueous phase. So this is one where we have the self-assembled micelles, self-assembled nanoparticles on polymeric micelles like this. Now, which one we have to choose for a particular catalytic effect? It all depends on the reaction condition. If the reactant A plus B giving rise to C, and you want to facilitate this reaction, and you want that A and B should be facilitated to convert into C, and the gold nanoparticle act as a catalyst, and you will add A and B solution, expecting the reaction is going to be facilitated. If particles are going to catalyze the reaction, that's fine. But the thing is, if A and B, they are aqueous phase, solubilized in the aqueous phase, unable to approach the gold nanoparticle because of their solubilization properties, then you have to find out another way. How can you allow the A and B to see another site where they can undergo the catalytic reaction? I mean to say that if A and B are possessing some kind of amphiphilic properties, as I told you, amphiphilic property where you have polar and a non-polar functional group, right? So then A and B, or only A or B, both may not have amphiphilic molecules, they would prefer the micelle. They will absorb on the micelle because micelle itself is an entity which can solubilize, as I told you. That micelle has the ability to solubilize any kind of solute which is either the polar or the non-polar. Any range of polarity of the solute can be solubilized in the micelle. The polar will be adsorbed on the surface, non-polar will go into the core. So therefore, micelle is a unique kind of a reaction vessel or a sphere or a ball which can accommodate any kind of a molecule. So these reactants will be accommodated in higher concentration in the micelle. Okay. And when they are in the micelle in higher concentration, gold nanoparticles are already there. The reaction which we are saying A plus B giving rise to C is much more facilitated. If I'm saying reaction vessel 2, reaction vessel 1 or 2, the reaction 2 is much more facilitated in comparison to reaction 1 because we have created a template which is not only the platform for the self-assembly of so many gold nanoparticles, but it also attracts the solubilization sites of the reactants whose reaction we want to catalyze. So therefore, individually, the gold particle effect may not be that enough, but they are in a large concentration over here, and they can facilitate the reaction to a very high degree of completion. So this is the main advantage of such kind of assemblies, and people are doing studying the several catalytic effect on the self-assembled state of the gold nanoparticle on micellar assemblies. So this was the advantage of, I mean to say, for the templatic effect of the micelles in order to accommodate the gold nanoparticle and a really good substrate for any kind of catalytic effect. Now I'm going to take you to another kind of nanoparticle in self-assembled state. This was gold nanoparticle, right? Because we have learned about the reaction for in atomic state, starting the crystal growth, nucleation, eventually ending up with the formation of nanoparticle. Here we are going to talk about the silica spheres. What are the silica spheres? You see such a fine assembly of nanoparticle, right? Here, where is the scale actually? Scale is over here, right? So anyway, so scale is over here. So what is size? Usually silica spheres have a size quite big, from nanometer to all the way up to 1 micron. But most of the time we are using silica spheres close to 200, 300, or 400 nanometer size. So you may assume that this is of that much size. What is the meaning of silica spheres? So what are they? They are in fact the silicon oxide. their actual composition is silicon oxide and other derivatives, you may say other alkyl groups associated with this. But how they are produced? There are few reactions which I have shown over here. This is the one hydrolysis in the basic medium. So if you are going to take this molecule that is the derivative of silicon oxide, and you treat it in the aqueous phase in the presence of the basic medium, you can produce the hydroxide, right? And that hydroxide can be further used in a polymerization process, which is known as condensation, and eventually leading to the formation of silica spheres. This is one way. The other one is the other way is you can take tetraethyl orthosilicate, ethyl. So it is CH2, CH3, CH2, CH3. There are four tetra, 1, 2, 3, 4. Tetraethyl orthosilicate. So it is the salt, it's a compound of silicon. So this is the name of this compound. When you are going to treat this compound in a medium of ethanol plus water, facilitated by ammonia, and the condensation of the seeds are going to take place. So it is going to grow exactly the same way as we were talking about gold atoms. So it is base catalyzed hydrolysis, right? And slowly the nucleation sites, I mean to say that so they are going in a network structure, uh, polymeric structure to create a bigger and bigger surface. So, and you see this is the first step, condensation of the silica seed, and that keeps on going till uh, till we continue the reaction. Eventually, we stop the reaction and we have the silica sphere of the required dimension. If you want the smaller silica spheres, obviously you have to stop the reaction in a shorter period of time. But if you want bigger, you let the polymerization to happen, condensation to happen. Eventually, you have the bigger silica spheres. And you see, they are means a controlled reaction condition can produce the silica sphere of very narrow shape variation, and they are all of the equal shape and size. Now, this is the way we actually create silica spheres. If you ask me where we are going to use it, most of the your sunscreens, they are having the silica spheres, silicon oxide components, right? Apart from that, there are very important applications for these nanoparticles. That is the template effect. You see over here, template effect. What is the meaning for this? Now, suppose if I have uh, the micelles of SDS or CTAB, as I mentioned earlier, that you could have the micelles of CTAB or SDS with much smaller size in comparison to that of the polymeric micelles. Okay. Now, the thing is that how can we use them as template? Here we are using the micelles of conventional surfactant, SDS and CTAB as the template to create the sheets of silicon oxide. You see the sheets of silicon oxides with the pore size 1.5 to 10 nanometer. So this is the pore size. How can we prepare this? The condensation reaction which you have seen over here, if you are going to conduct on a template, you can create such assemblies, right? First, we are going to talk about this. In order to create this kind of silicon oxide structures, we are using the micelles template. What you do, you have in the beaker you have the micelles, okay, the micelles over here, and the reaction which you are talking about over here, this one, this reaction is conducted in the presence of the micelles, these micelles. So when the silica spheres will be created, they will absorb on the surface of the micelle. The seeds are going to absorb on the surface of the micelle, and if you keep on conducting the reaction, the seeds will merge and create a sheet. This is a sheet of silicon oxide. Uh, uh, the way you have the silica spheres, the same way you have the silicon oxide that molecules, condensation. They, the micelles, so this one is silica seeds. They are going to merge and create a shell. If you keep on growing this shell, now the shell is grown, right? And eventually you have another micelle over here and you have another shell, right? The shells are going to merge with one another, creating a three-dimensional structure, the one you see over there. This structure I'm talking about. And you see, these are the different micelles. So these different micelles, they merged together, created a template, and that template eventually created this kind of oxide, silicon oxide structure. Now, ask me that how, where are the micelles then? The micelles are here, and where are the micelles? These are the, because if we have to use the silicon oxide pores, empty for a certain application, we need to remove the micelles. What you do after the completion of the reaction, you take the sample, put it in the furnace, and heat it at the elevated temperature. Carbon is going to burn away, right, or decompose. So the micelles are going to decompose, and with the result of which you are left with simply this template of silicon oxide. This is the way we grow it. This is one way of creating the templated structure of silicon oxide, and we can use it for some purposes. So I'm going to talk about that as well. Let us assume that we want to create the hollow tubes of silicon oxide. These are the hollow tubes. You see, hollow tubes. It's completely. It's only that of silicon oxide uh, 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 kind of the molecules, right? And they are empty. And this is the image. These are the tubes. They are hollow inside. But the question is, create. So they are created as we created over here, but instead of the micelles, the conventional surfactant, here we have to take the polymeric micelles, which have the shape transformation. Remember, I told you that micelles are going to transform their shape if the if we increase the concentration of the surfactant, and the micelle is going to take this shape. So there, so now here are the surfactant molecules, and this is the elongated micelle. So elongated micelle, right? And the shape transformation, this is sphere, and this is, you may say it's rod or a cylindrical micelle. If I take such cylindrical micelles and allow the reaction which I have conducted over here to happen, so I can create a sheet of silicon oxide, derivatized seeds over here. So now this is the same thing which we created over here. It is the same thing created. We have created on the spherical micelle, but here we have created on the cylindrical micelle, right? So it means that now the cylindrical micelle is the template. Here the spherical micelle was the template. Spherical micelle helped us to create this kind of structure with the empty pores inside, and the polymeric micelles, cylindrical micelles, created this kind of structure which is coated with the silicon oxide, and eventually when we treat in at elevated temperature, we will decompose the micelles, and hence we will produce the empty tubes. Now these empty tubes you could see here clearly in this image. This one is the scanning electron microscopic image, not transmission electron microscopic image. So these are the tubes. You see so many tubes are here. So once you have created such kind of assemblies, it is up to you. There are plenty of applications for them. This can act as a filter for any kind of, usually the filter paper which you use, even the Waterman filter paper, very fine filter paper, the pore size is in micron. You never reach this pore size in nanometer, right? So therefore, there is no way you can have the filter paper with this uh, 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 this much dimensions. The reason is that this is the only way, means this can be achieved through the synthesis, not the mechanical process. There is no way you can convert small pores through the through any kind of uh, uh, I would say mechanical processing. It's not. It is through the chemical reactions only, because the chemical reactions involved atoms. So you control the growth or the reaction, eventually ending up to any kind of pore size or the morphology ranging from nanometer to micron or any other. If somebody asks you, what is the reason for that? Suppose I have a scissor, right? And I have to cut a piece, right? The scissor cannot cut a piece thinner than its blade. Am I correct? Right? If the scissor blade is already in micron, there is no way you can cut any piece smaller than the micron. The same kind of argument we can apply. So therefore, why chemist or the chemistry is that important for any kind of research related to the material science? Because it is the chemist who knows how to use the different kinds of molecules or the self-assemblies of the micelles of different dimensions for the growth of any kind of other species. So I mean to say that when you start with the atoms and the molecules, eventually you can end up with the morphology with the pore size in the nanometer range. But if you are going to start with any kind of mechanical device, which is already existing in the micron or bigger size, there is no way you can create a nanometer object. So this is the difference between the mechanical process and the synthetic process. If somebody asks you, why would you prefer the uh, 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 down up approach rather than the top down approach? Top down approach is grinding the material to a smaller size, but bottom up approach is starting with the atoms. So the thing is that starting with the atoms, atom is the smallest entity we have. Am I correct? Atom is the smallest building block we have. Any form of the matter which we have here, either in the solid state, liquid state, or the gaseous state, that is being created by the building blocks of atoms. So therefore we cannot slice the atom. There is no way. So any entity which cannot be bifurcated, broken up, so that is the smallest and the building block. So therefore when you start with the building block whose size is less than 1 nanometer, obviously you can achieve any object bigger than that. And that is the reason when you start making a room or a house, you start by using the bricks, because your ultimate aim is to create an object which is much bigger than a brick, right? So it's the same kind of philosophy we are going to apply for the bottom-up approach, because the bottom-up approach provides of numerous kind of possibilities to create a nanoparticle or nanomaterial of any dimensions, because we are starting with the atoms. So that is why here you have forces in the nanometer range, and is you have the tubes, silica tubes. So we are going to see that how can we use them. Okay, now I'm going to, as I told you that we have created this. So this was the template, right? And it's the same template which I mentioned in the previous slide over here. Template. I mean to say that if you keep, if, if, uh, if you take a high concentration of the micelles, you will have several interconnected templates like this. And this is the three-dimensional structure. You see, this template, silicon oxide, exists in this network structure. You see the pore size, right? This is the three-dimensional network structure. And the most surprising uh, 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 aspect of this is, read this. You take just 70 milligram of this sample, 70 milligrams. You could imagine that it's so tiny sample. Surface area it is possessing is equal to the one bedroom apartment. Surface area. The one bedroom apartment has plenty of open space. Suppose if you ask me how we can compare this, that is, it's not the size I'm talking about over here, right? So it is the empty space. There's plenty of empty space over here, but interconnected three-dimensional area is occupying that empty space and hence creating a much bigger surface area that is equivalent to this, right? What is the advantage of this? Now you see that, you know, the while drinking tea, you want to remove the tea leaves by using the sieves, right? And it is up to you that what is the mesh size, the bigger or the smaller. The smaller the mesh size, so even the small particles of the tea leaves, so they are going to be filtered out, right? So the same way, if you are having pore size much smaller, you want to use this, you cleaning the drinking. So this is the best kind of network structure in the nanometer range, with size pore is so tiny that it has a very high affinity to clean arsenic from the drinking water. Arsenic poisoning, the heavy metal poisoning, is highly toxic and it's cancerous, you know. And therefore, this kind of structure actually has, apart from other application, I am just, because the easier way to understand that what kind of application we are going to use for this kind of material. So it is the cleaning of the contaminated water with arsenic. Apart from this, you can use it for catalysis, fuel cells, and the solar cells, because the silicon oxide and your solar cell composition most of the time, so they are the silicon oxide nanoparticle. So there are plenty of applications. So I mean to say that advantage of what I have shown in these few slides, I have just covered 6 slides. I have I have mentioned that the advantage of the templating effect. If you have a clear templating effect, you know how to create the templates and what kind of assemblies you are going to use as templates in order to create the network structure, then there are plenty of opportunities in the material science. Okay, now we are heading another kind of bioapplication. You know the silica spheres. So these are the silica spheres. If you simply, for the time, yeah, over here, this is a scanning electron microscopic image. You see the fine silica spheres, clear 200 nanometer size. Approximately 200 nanometer size dimension, the diameter is close to that. Right. So this is the scanning electron microscope image, SEM, but this is the TEM. TEM can look through the object. So therefore, they look transparent, because they are not metallic, they are oxide. So therefore the diffraction is low and hence they look semi-transparent. You see the boundaries over here clear, but the dark particles from where you have a much more diffraction of the electrons. So those dark particles, nanoparticle, if I carry out the gold nanoparticle synthesis in the presence of these spheres, I can grow the gold nanoparticle on the surface of the silica spheres. What is the advantage? Again, I told you that there could be the advantage in the catalytic effect. So the silica sphere has the ability to absorb or to accommodate. It's a porous structure and to absorb the reactant molecules. Once they are at the reactant, once they are in the silica spheres, then if the gold nanoparticle surface is the catalytic surface and it can facilitate the catalytic reaction. Here, what they blood, you can use them in the presence of blood. So these are the silica spheres. Now you will be wondering that it is 200 nanometer, but here when we are going to place these two on the surface of the blood cell, they look so tiny, only tiny dots, because look at the scale bar. It is 2000 nanometer, am I correct? 2 micron. 2 micron is 2000 nanometer. It is 10 times bigger than this. 200 nanometer, 2000 nanometer. So the blood cell size is much bigger that resides in the micron scale, micrometer scale. So therefore, there is a huge size disparity between the gold nanoparticle or the silica spheres and that of the blood cell. You can use them as a drug. Right. So here you see uptaking of the silica sphere by the by the animal cell. You see, first the silica sphere in low resolution attached to the surface. Slowly it enters, it is taken up and it is already inside the cell. Right? So in this way. Now what is the meaning for this? The meaning for this is that if it is carrying drug or any kind of bioactive molecules which you want to use inside the cell assembly, right? And you want that it should be released inside the cell, so you need a carrier. You need a vehicle that can carry the load, a truckload of some amount of the substance, and that truckload of that truck will act as a vehicle that will carry it and enter into the assembly where it is going to deliver it. So that is the purpose of this. So there are many biological applications. So I would say that just look through the publications where you have applications of the silica sphere, and you will find many interesting applications probably on which you start your next research project. So how to become a scientist? This is a very important usual question. Many of my students are as well as the other students, so they ask me, I said, always start with a small thing. Just look in which direction you want to work and go through a publication, pick the idea, and see that how can you start some kind of preliminary experiment. And you start just preliminary experiment slowly, because among all the living organism, intelligent species is the human being. So God has provided you the brain. Eventually, you will see you have new ideas coming up. What if I'm going to take this reaction in this side, that side? That is the beginning of a good scientist. Anyway, going ahead, we are left with few minutes. So I have few more slides. Just now. Yeah. Okay, so I have important slides because now I am taking you to directly the biological applications. Okay, this is BSA, bovine serum albumin, right? BSA, you are very familiar. This is the three-dimensional structure, domain 1, 2, 3. This is the globule, BSA in its native state at 40 degrees centigrade, which we say a room temperature. If you increase the temperature, the protein is going to unfold like this. This is unfolded from denatured protein, 80 degrees centigrade. Can we use the protein to produce biodegradable films? Now you know that we have strict rules and regulation not to use the plastic bags. And you will see that most of the bag, the written biodegradable bag, right? So is there any way that if a protein is freely available, I think you have plenty of proteins, gelatin is freely available, right? And in this way, you can use the protein to produce some kind of film that can be used for any purposes, for biodegradable purposes, for carrying bag, or for any kind of biological activity. So see over here, how can we do that? Gold ion in the presence of BSA at 80 degrees centigrade, it is possible to create a film. This film is created now, but you have to be very careful in arranging the mole ratio. When the mole ratio is less than 160, you cannot create the film. When the mole ratio is 167 or higher than this, then the film is created. This film is the protein film. But you see the dots created over here? The dots are in fact the gold nanoparticle. So dots are providing the reinforcement, the strength to the film, right? Otherwise, the film is brittle and hence it may not be useful. But when you use the gold nanoparticle act as a binder. So hence, the film is quite flexible and where is that? Just a minute. Okay. So what we do, we can choose BSA. Sorry about that. BSA is very expensive protein, you know. How can you use a BSA for this kind of purposes? But on the other hand, Zein protein is very cheap. It's a corn protein. And actually, we explained the zein protein. It's very cheap, freely available because in North America we have plenty of corn. The way you have plenty of wheat and rice, but in North America when you drive across the cities from one place to you see very big corn fields. So corn is the cheapest crop in North America, one of the cheapest crops in North America. And the zein protein is a corn protein. So, but the problem is what insoluble. It's non-toxic, biodegradable, easy to process, highly brittle and less flexible. And it is already frequently used because the non-toxic is used in few food packaging material, which is biodegradable, and chewing gums. The most of the chewing gums which you use every day. So zein is a part of that, and in pharmaceutical medicines. So it has something this kind of a structure. The protein, you don't need to fully understand that what is the meaning of this. It is the same way as we explain the BSA three-dimensional structure. The BSA three-dimensional structure was right. The same way the three-dimensional structure of zein protein is cylindrical structure. We call it cylindrical structure. Since it is highly hydrophobic, so the zein protein contains predominantly hydrophobic domains. So what we do actually, since it is not soluble in water, instead of dissolving in water because it's not possible, so what we take the solution. We take the surfactant solution, micellar solution. We we dissolve zein, and in this way we prepare the zein solution. Now zein is dissolved in the micellar solution, producing a clear solution. What we do actually, we use the zein protein the way we use the BSA to create the film. We can also use the zein protein. So once you have created the aggregated assemblies of BSA and gold nanoparticle to produce a big sheet of biodegradable film, we add the zine because zine is much cheaper. We add the zine over here, and in this way we create a sheet. You see yours? This is the sheet, and it is shown in the folded state by forceps, right? So now this is made up of proteins only, but along with the proteins you have the gold nanoparticle which help to maintain the strength and the flexibility. So where you can use it, so this is the tensile strength and strain at failure. So these are the experimental data. So how strong this system is. And, but for any kind of food packaging and other kind of a packaging where, so when this film is going to decompose, protein is instantaneously going to be decomposed or taken up by the insects or the yeah, available in the soil, and the gold nanoparticle, because the metallics, so they are going to be disposed of anywhere. So in this way, what I say that this is one of the best example of the protein films which can be used for any kind of purposes as degradable films. So hence to solve the environmental problem. So here we have uh, even cheaper. So you can use starch. Starch is really available, right? So here we have prepared the films by using starch and glycerol. So starch and glycerol. So the glycerol coated cadmium sulfide. If you want to create a colored film, so here we have taken the cadmium sulfide nanoparticle, and here we have taken the gold nanoparticle. You see the color. The color of this film is slightly pink because of the gold nanoparticle. This one is yellow because of the cadmium sulfide. So cadmium sulfide electronic properties are much different from that of the gold nanoparticles. So it's not only, I'm saying that it's not only as a packaging material, but apart from that, suppose if you want to study the electronic property in some other sense, then you can also perform that kind of applications as well. So eventually, unfolding of proteins in the presence of nanoparticles can lead to the formation of structures, protein films, which, which have the wide applicability everywhere. Nanoparticles promote structural enforcement in biomaterials. So why we are using nanoparticles? Can't we use the simple proteins without the nanoparticles? The thing is, yes, you can use it, but the thing, by adding nanoparticle, it is providing the structural enforcement. So therefore, you may have the better applicability when you are using the protein films embedded with nanoparticles. Thank you so much for your attention. Any question? Just announce that the next